Okay, video. This title slide is transcription and translation. We are just going to be covering transcription in this video. Sorry, I did not update my hedgehog photo. We'll get there. I'm kind of scrambling to get this recorded here, but let's jump in. So, the next bit of this class is going to be focused around this idea of the central dogma of biology, where information, genetic information, flows from DNA to RNA to protein. That's kind of the three main players. Essentially, we have DNA, we make RNA off of that DNA, and then we make proteins using that RNA. So there are two main processes in this. Transcription, which we're going to be talking about today, and that is the making of RNA from a DNA template. It's a lot like DNA replication, except you're just making a single RNA strand instead of two DNA strands. And then translation is going from RNA to protein. So the way I had learned this in high school was transcription. If you transcribe something, you're kind of just like copying it from one place to another. It's the same info, but you're just copying it from online onto your notebook or something. Whereas translation is actually going to a different language. So in this case, the language we're going from is the DNA, RNA, nucleic acid language to the protein language. And that's what translation will be. We'll be talking a lot with messenger RNA. I just wanted to make sure you have that abbreviation. And remember, ribosomes are the sites of translation and protein synthesis. So translation itself, and we are going to go over this more in class, by the way. Transcription is directed synthesis of RNA. So it uses an RNA polymerase. It does the exact same thing as DNA polymerase and has the exact same restrictions, except that it just makes RNA instead of DNA. But it can still only add things onto the 3' end of the growing strand. Base pairing rules with RNA are basically the same. Cytosine pairs with guanine. The difference is that RNA does not have thymine. It does not have the T. So instead of adenine pairing with thymine, it pairs with uracil, just a different nucleotide. There are three main steps: initiation, elongation, and termination. We are going to start with the initiation process. So in the initiation process, there is a special DNA sequence ahead of a gene called a promoter sequence. And this is where RNA polymerase actually attaches before it actually starts to make the gene. So the promoter sequence isn't the gene itself. It's just indicating kind of the start point of the gene. And then in eukaryotes, it's a string of just adenine and thymine all together. That is called a Tata box, which is one of the weirdest names in bio, I think. And then there are also these proteins around called transcription factors that just help RNA polymerase attach. They just make it easier for it to happen. So first thing that needs to happen is RNA polymerase needs to attach to the DNA strand at the promoter sequence. We're going to be talking about RNA a lot in this unit, and there are three main types and three main functions that you need to know and keep in your head as we go through this. First is messenger RNA. That is what carries information from DNA to the ribosome. That's what we're making in transcription. tRNA is translation RNA or transfer RNA, and it is the RNA that brings amino acids around. It'll play a bigger role in translation, which we will talk about next time. And then rRNA, ribosomal RNA, is just RNA that composes part of the ribosome. It's kind of the attachment point for the mRNA strands that will come in. So those are our three types of RNA. We're mainly going to be focused with mRNA today. So as we know, transcription factors are proteins that are just going to help RNA polymerase attach to the DNA strand, which helps us kind of regulate what DNA strands actually get transcribed and what genes we actually make. So that's the first step. Second step is elongation. This is just RNA polymerase adding in all the nucleotides off the complementary base pairing. Yet again, it is to the 3' end of the growing molecule. We do not have a leading and lagging strand in this case because only one of the two DNA strands is actually being copied and made into RNA. It is called the template strand. That's the one that actually gets copied and read. There's a non-template strand, which is whatever is bound to the template strand, and that one is not read or copied. So a little more on these. You'll see many different names. You'll see the coding, sense, or non-template strands are the ones that are not being copied. Wait, do I have this backwards? No. And then we have the non-coding strand, antisense strand, or template strand. All the same terminology. And this is what actually gets transcribed by RNA polymerase into mRNA. So 3 different names, just be aware that they're all there. Then finally we get to the end of transcription. It is the termination process. So there's a specific sequence in RNA that's just basically a stop sequence. And so when it hits that specific RNA sequence to stop transcription, RNA polymerase detaches, and we have our, technically we have a pre-mRNA molecule. Because after transcription in eukaryotes only, if we were a prokaryote, this is where we would stop, but eukaryotic cells do a little bit of post-transcriptional RNA processing. So there's a few main things that happen here. One, they put a cap on the 5' end. It's a methyl cap. It just kind of protects the mRNA. And then the 3' end gets a poly A tail, just a bunch of adenines strung together. We'll talk in class maybe about why we might have those. And then we have the introns and exons. So you may have seen those boxes at the back of my room. Introns are non-coding regions of the mRNA strand. They might be used sometimes, but they are cut out of the mRNA strand as part of processing. And then we keep the exons. Exons are the coding regions, and so we keep them and splice them together. Splice means join, not cut, by the way. People often make that mistake that splice means to cut. It actually means to join. And so there's this idea of alternative splicing in eukaryotic cells where after we make the gene, we basically have, or our cells at least, have the ability to decide what is an exon and what is an intron. It depends on what they want out of it. So you can get kind of different versions of mRNA out of the same gene. This is used in our immune system with antibodies. We basically just mix and match all the introns and exons, and it gives us a bunch of different antibody shapes. So rather than having a million different antibody genes, we can have a few and just mix and match them. So that is alternative splicing. And that is transcription in a pretty fast time scale. We will talk about this in class. Translation will be next time. Enjoy. Hope you had a good weekend.